Welcome to the School of Theoretical Modeling webinar, Helical Assemblies. We will discuss today structure and function of protein helical repeats. Helical assemblies are important components of many organelles: inflammasome, proteasome, apoptosome, spliceosome, intersome, and many others. Helical assemblies are implicated in pathways associated with many diseases, including cancers. Ankyrin repeats for biofuels, design ankyrin repeats, DARPins in immunotherapy, ankyrin repeats for crystallization. These are just few of numerous applications of this remarkable molecule. New studies of structural determinants of helical assemblies, chirality of helix-helix interactions, and implications for the assembly of helical rows are major novel approaches that allow reliably predict three-dimensional structure. Structure modeling of new ankyrin repeats proteins and their preliminary structures, candidate proteins for structure prediction, and how to apply structural modeling in biomedical research are our major topics. Pathogenic microorganisms present a danger and are recognized by the organism's innate immune system. Inflammasome multiprotein complexes govern the innate immune response. Structural studies of inflammasome molecular complexes reveal details of the assembly of these multiprotein bodies. Leucine-rich repeats and nucleotide binding domain are major components of inflammasome assembly. This nucleated polymerization process, with involvement of ATP, contributes to our understanding of enzyme activation. The activation mechanism is self-propagating. Two receptor molecules control the process of polymerization. It starts with one molecule binding microbial component and triggering assembly of 10 to 12 other molecules into allele structure. This signaling complex is activated by infection, metabolic malfunctions, and other signals. Ankyrin repeats are scaffold proteins or adapter proteins. Ankyrin repeat domains are found in hundreds of proteins. They are protein-protein interaction modules that bring together many components in clusters and assemblies of various shapes. Ankyrin repeats connect membrane proteins, ion channels, receptors, cell adhesion molecules to cytoskeletal proteins such as spectrin. Ankyrin repeat proteins target other proteins to plasma membrane. Conserved overall shape of the molecule, but unique surface formed by its functional groups allows recognition of multiple targets. Ankyrin repeat and sterile alpha-motif domain containing protein has 7 ankyrin repeat units. Two isoforms, 1A and 1B, show similarities as well as dissimilarities. ANK-S1A in colorectal cancer cells is SRC family kinase substrate and links EGFR, PDGFR, EphA8 to downstream signaling cascade. Its ankyrin repeats domain is important for assembly of signaling complexes. This adapter protein, as mediator of assembly of multiprotein complexes, interacts with multiple proteins. ANK-S1B is major component of postsynaptic density, where its phosphorylation by ChemK2 mediates molecular reorganization. Interactions of ANKS1B with CREAT1 through PTB domain affects endothelial permeability. ANKS1B is involved in regulation of subunit composition of NMDA receptor, affects synaptic plasticity, acts in renal carcinoma and other processes. Other proteins for which preliminary structures were obtained include tumor suppressor, HECT, and ankyrin containing protein, NYPR1 that contributes to cancer, lysozyme-like protein, LYSL5, and ankyrin A. Among candidate proteins are ARF GTPase activating proteins, GIT1 and GIT2, NRARP that is involved in Notch signaling pathway and functions as adapter protein that brings protein complexes together through protein-protein interactions. Also, ankyrin domain family members that are implicated in many cancers, including breast, colon, prostate, ovary, and pancreas tumorigenesis, as well as non-small cell lung cancer, are important candidates for structure determination. They are known as cancer biomarkers and are promising for vaccine development. What are major components of the prediction method that allows to generate reliably three-dimensional structure of the helical assembly? Chirality of helix-helix interfaces determines direction of the assembly. For each type of assembly, pattern of contacts is unique. This pattern of contacts on the surface of the alpha helices determines how they fold. Orientation of helices affects ligand binding. Conformation of helix-helix interface determined by specific amino acid combinations at helix-helix interfaces. Correlates with ligand conformation. Involvement of helical edges is key to the type of the fold. Protein fold depends on what helical edges are involved in helix-helix interface. Specific amino acid combinations of helix-helix interfaces are recognition units. Whether one of alpha helices binds another alpha helix is determined by these specific combinations. Various types of specific pockets exhibit specific amino acid combinations. The coordinate system of the layer allows us to compare specific pockets. It also is very convenient system to observe the direction of the assembly of each type of assembly. We can easily see the rows of helices that are or are not superimposable on each other. There exists a large gap between data on primary structure of proteins and data on secondary and tertiary structure. Theoretical models close this gap and give researchers structural data that are invaluable for understanding principles of protein structure, understanding cellular pathways, and drug action. They save time and resources. They are particularly useful with emerging growing amount of data on individual genomes and personalized medicine approach. With improvement of methods and quality of structures, more and more data become available.